In this video, I discuss several applications of silver nanoparticles for antimicrobial applications and also compare their antimicrobial activity to that of pharmaceutical antimicrobial treatments. If you enjoyed watching this video, click on like, subscribe, the bell, and share so that you can be notified of future videos that I post in the channel. So the question is, what are antimicrobial agents? They consist of materials that are used to treat and prevent infectious diseases and include antibiotics, antivirals, antifungals, and antiparasitics. There are many pharmaceutical drugs that are used as antimicrobial agents, but one major problem is that many microbes, such as bacteria, have become resistant to many antibiotics. Hence, there is a great need to develop new types of materials that can be used as antimicrobial agents. Silver nanoparticles are very promising to use as antimicrobial agents to overcome the resistance of antibiotics, antivirals, and antiparasitics. Polyethyleneamine capped silver nanoparticles are used as antimicrobial agents, in which they are tested for cell death of E. coli bacteria. The use of these nanoparticles to assess cell viability is discussed in the next few slides. This figure shows a comprehensive characterization of the polyethyleneamine capped silver nanoparticles. Panel D shows the TM spectrum of the silver nanoparticles, and Panel C show the dynamic light scattering data, which can be used to determine the size of the silver nanoparticles. Figure A shows the surface plasmon band position of the silver nanoparticles, and Panel B shows the excitation and emission spectra of the silver nanoparticles. The polyethyleneamine capped silver nanoparticles were used to assess the percent age cell viability as a function of the concentration of the silver nanoparticles. It can be seen that there is a dramatic reduction in the percent age cell viability starting from 50 nm of silver nanoparticles and continuing onwards to 500 nm of silver nanoparticles. This shows that the silver nanoparticles are very effective in preventing or drastically reducing cell growth over time. The process of hemolysis involves rupturing of the red blood cells. A hemolysis study was conducted using the silver nanoparticles to assess its biocompatibility. The percent hemolysis was plotted as a function of the concentration of the silver nanoparticles. From the graph, it can be seen that at the highest concentration of the silver nanoparticles, which is 500 nm, there is only 2% hemolysis that occurs, indicating that the silver nanoparticles are highly biocompatible. Panel A shows E. coli samples in which the cells are alive and emit green in the 490 and 515 nanometers region. Panel B shows E. coli samples in which it can be seen that there are many dead cells which emit red at 535 and 617 nanometers. Panel C shows Panel A and B merged together. A systematic study was conducted to investigate the effect of the culture conditions on both the stability of the silver nanoparticles as well as their antimicrobial properties. The UV visible spectra were obtained for the silver nitrate compounds as well as the silver nanoparticles that are synthesized. It can be seen that there is a surface plasmon band in the case of the silver nanoparticles. The UV visible spectra were obtained for the silver nanoparticles in the presence of different culture media at 0 time and after 24 hours. In the case of when the silver nanoparticles were in the presence of the culture media at 0 time, there were minor differences in most of the culture media. One exception is that in the presence of YPD culture media, there was a noticeable decrease in the surface plasmon band. In the spectra obtained after 24 hours of incubation in the presence of the culture media, there was a lot of variations in the surface plasmon band that was observed. This suggests that the physicochemical properties and stability of the silver nanoparticles were affected. The redshift in the surface plasmon band lambda max position suggests that there is precipitation or aggregation of the nanoparticles after 24 hours. The silver content in the presence of the 3 different culture media at 0 hour and 24 hour incubation was investigated. It was observed that the RPMI culture media resulted in the lowest silver content after 24 hours. It was observed that in the case of YPD culture media that the silver content increased after 24-hour incubation time. This suggests that silver ions are more readily released in RPMI and MH culture medias compared to the YPD culture media. The effect of the concentration of different components of a cell culture media was investigated. It was determined that 1.75% peptone results in the maximum amount of E. coli growth. Also, it was determined that 0% dextrose resulted in the maximum amount of E. coli growth. The reference media used as a control for maximum growth of E. coli contains 1.75% peptone and 0% dextrose. A systematic study was conducted on the use of a variety of different culture growth media to determine the media that results in the maximum percentage of growth of the E. coli bacteria. It was observed that using the TSB growth media resulted in the maximum growth of E. coli bacteria. The antimicrobial activity for E. coli was conducted using the different culture media, and it was determined that the lowest microbial activity occurred when RPM culture media is used. The highest mic value occurred when the TSB culture is used. It is also worth noting that the antimicrobial activity can vary up to 2 orders of magnitude depending on the culture media that is used for the studies. Overall, the big problem is that many kinds of bacteria are resistant to pharmaceutical antibiotics. As a result, silver nanoparticles are promising alternatives to antibiotics for bacteria such as E. coli. If you enjoyed watching THS video, click on the like, subscribe, the bell, and share to be notified of future videos that I post in the channel.